I'm Na from Helmholtz Munich. I'm a CGSI regular. I've come twice as a trainee, and this is my second time as faculty. And today I'm going to be talking about depression. So how many of you were in Jonathan's talk a few days ago? Okay, so not everyone. I'm gonna repeat some of the stuff that he talked about, and this is so that I set up the stage for what I want to be talking about today, which is this problem of that we face in genetics of depression, which is the trade-off between power and specificity. So the problem with depression is that it's badly diagnosed. It's badly diagnosed in the clinic. It's even more badly diagnosed if you had a self-report questionnaire doing the diagnosis for you. So the best diagnosis that you can make for depression is based on structured clinical interview, and even that has quite a high variability in practice. This has in terms of both false positives and false negatives. So for a person who wants to do a genetic study on depression, the best thing you can do to overcome this variability in diagnosis is to perform a standardized deep phenotyping interview on everyone that comes through into your study. And in this way, you can minimize misdiagnosis and also maximize the collection of data of their clinical characteristics. The problem with this is that it's time-consuming and costly. And usually that limits the sample size that each individual cohort is able to collect. So this is very early GWAS on depression. There were many small cohorts collected by various places, each in the range of hundreds of thousands of samples. And in 2013, the Psychiatric Genomics Consortium put them all together into a meta-analysis, totaling around 9,000 cases and 9,000 controls. And this still didn't give a GWAS significant hit. So this was despite depression having a substantial twin heritability of around 40%. And that's similar to some of the other diseases that GWAS has started to give significant GWAS hits at this point in time with similar sample sizes. So that was disappointing. And, but that could also be because the cohorts were collected in various places. They're from different populations. Maybe slightly different diagnostic questionnaires were used. So maybe they were still too heterogeneous for this to work out and too noisy for this to work out. So then came Converge, which is the cohort that Jonathan Flint and Ken Kendler collected in China. This is the largest hospital-based study on severe and recurrent depression. And we have 6,000 cases and 6,000 controls. And deep phenotyping was performed on every individual by trained clinicians. Somebody at the back? Okay, I'll try to be even louder and see whether this works, but I may not last very long. So as I was saying, this is the most stringently phenotyped data set to date. We have 5,000 cases and 5,000 controls, and it's likely the one with the lowest misdiagnosis rate. So it was hypothesized that with this kind of deep phenotyping and stringent data collection process, it would reduce noise and heterogeneity such that there is enough power to find GWAS hits at this type of sample sizes. And this hypothesis panned out for us, and we were able to use this data to find the first 2 GWAS hits for depression ever. But it was clear to us, that this was the right strategy. We should be doing deep phenotyping, we should be cleaning up the data, and that would give us power in GWAS. But it was also clear to us that the sample size just needs to get much bigger for this to get anywhere close to explaining the genetic variability in depression. So in short, we need bigger sample sizes. And I think for a while, what we thought was that that would mean more funding for deep phenotyping. So we'll see a nice arrow going from needing higher N to more deep phenotyping. But instead, there's a different approach, which is to get bigger N through quicker methods, such as shallow phenotyping. And by shallow phenotyping, I mean assessing whether one has depression using an individual's question to one or two simple questions, such as, have you ever been diagnosed or treated for depression? And this was the approach taken by a company called 23andMe. It's a consumer genomics company. And in 2016, they were able to use a single question to get data from 300,000 individuals on whether they have been previously diagnosed or treated for depression. And that gave us 17 GWAS hits in 2016. The second way is to pull ICD-10 diagnostic codes for the disease from electronic health records. And these are doctors' diagnoses of individuals who came into the clinic. So many groups have done this, and the Psychiatric Genomics Consortium has also pulled these together with data from 23andMe and other cohorts to do large-scale meta-analysis from a range of different types of data collection processes. And as you can see on this slide, the number of GWAS hits on depression defined in these ways have grown very quickly over the years, and it has resulted in massive increases in number of GWAS hits found. The issue with this, however, is that with high power, this comes at a cost of specificity. And a couple of years ago, we wrote a paper explaining this in detail, and I'll very briefly talk about what we did. So briefly, let's say the deepest phenotyping possible for depression is a clinically administered structured interview following the diagnostic criteria for depression. And on the other end of the spectrum, you have self-reported disease status. In the UK Biobank dataset, which is a 500,000 people dataset collected in the UK, there's a whole range of phenotypes that maps onto various points in this deep shallow scale. The deepest phenotyping done is through a self-administered online questionnaire on depression symptoms. And using this symptom, we are able to generate a depression diagnosis based on clinical criteria. And we call that lifetime MDD. And I'll call that lifetime MDD for the rest of this talk. The shallowest phenotyping possible in UK Biobank is this done through one question: have you been diagnosed? Have you gone to the doctor for depression? And we call that GPSI. That's the red thing at the end of that spectrum. So I'll call that GPSI for the rest of this presentation also. So here I show the GWAS on lifetime MDD on the top and GPSI on the bottom. And as you can see, the power is much lower for lifetime MDD than GPSI, as it has a much lower sample size. So the question is, can we use GPSI instead of lifetime MDD? Because it seems to give us so much more samples, give us so much more GWAS hits, seems like such a better idea. And we've done many analyses, and we're convinced through those analyses that the answer is no. So they seem to have partly distinct biology. And I only show two examples here. Firstly, the heritability of lifetime MDD is much higher than GPSI. And secondly, on the other side, the genetic correlation of GPSI with lifetime MDD is around 0.8. And that means the amount of genetic sharing between them is around 0.8 squared, which is 64%. And that's not a lot for two phenotypes that are supposed to be capturing the same disease. We've done many more analyses on this that went to show how shallow phenotyping, such as GPSI, gives genetic findings that are not specific to depression, but they capture spillover genetic effects from those of schizophrenia, smoking, neuroticism, etc., as a result of misdiagnosis. And all of this is now already published. In fact, 2 years ago, I have a talk at CGSI about all of these findings. It's on YouTube. And sadly, because of lack of views, as compared to other talks, it hasn't featured in Pageman's tribute to Big Bear talks a few days ago. So if you're interested, you can go and take a look. So I'm going to assume now that you're now all agreed on shallow phenotyping giving high power but low specificity. And I would argue that this makes them less desirable as phenotypes to be studying. But the problem is, with greater power in GWAS, it comes, it gives people greater motivation to get even higher sample sizes using this approach. And that closes that self-perpetuating loop. And what we end up is hundreds of GWAS hits, some more specific to depression than others. And there's no way of telling unless we do a complete separate analysis just to identify which ones are specific and which ones are not. And in some discussions with psychiatric geneticists who are very deep in that kind of analysis, they say that, okay, well, this could be done in knockdowns, can be done in cell lines. We could knock down all of those 300 hits in iPSCs and figure out which genes they affect, and through that we can look at biological pathways, etc. So essentially passing this problem on to our wet lab colleagues. But I think there's a few more things that we can do before we hand it over to them. So myself and a lot of collaborators, some of them who are in the audience, we think we can do a little bit more just on the data side also. So given we currently already have depression derived from deep and shallow phenotyping approaches, and we don't have a lot of deep phenotype, we have a lot of shallow phenotyping from EHRs and biobanks. And given that we have many more phenotypes in the biobanks and electronic health records that are correlated with depression, either through shared genetic effects or shared environmental effects, we can aim to better utilize both deep and shallow measures of depression in these cohorts to improve GWAS power while hopefully not compromising so much on specificity. And I'll tell you how we have approached this. So our first approach is to do this with phenotype imputation, which has been shown previously to greatly increase power. So first I need to show you how data looks like. So this is a cartoon of how data looks like in UK Biobank. Here we have lifetime MDD. It's missing in many of the entries, so a lot of people don't have this measure. We have a lot of shallow phenotypes of depression, as I've shown you on this, a few slides ago. They are slightly less missing than lifetime MDD. And then we also have other things such as other mental health measures and also socioeconomic status, comorbidity, education attainment, et cetera, et cetera. So in our study, we've taken not very carefully chosen 216 phenotypes that is supposed to be representing the depression relevant phenome. We then use a software called SolveImpute to perform a singular value decomposition on this phenotype matrix to obtain factors and weights, which are then combined linearly to obtain imputed measures for all of these missing values of lifetime MDD. And it also does fill in all the missings for all of the other entries also. So here I show two plots that show you how well phenotype imputation has done on the phenotypic level. The plot on the left shows you the R-square between the imputed phenotype and the copy mass observed phenotype. So at 80% missingness, this dot here, lifetime MDD shows a 40% increase, 40% R-square, and that translates into an effective sample size gain of around 150% if you look on the plot on the right. So this is our primary approach. And of course, nowadays, you have to do stuff with deep learning. So our collaborators, Sriram and Ozzy, who are not in, I don't know whether they're in the audience right now, they developed a deep neural network-based method called autocomplete, which uses an autoencoder architecture to try to achieve the same effects. So in comparing the results from autocomplete and SolveImpute at 1% and 50% copy masking in the input data set, autocomplete always does better than SolveImpute in terms of imputation accuracy. So as a result, we test imputation results from both approaches in all of our downstream analysis. So now that we have imputed lifetime MDD phenotype from the MDD-related phenome in UK Biobank, the question is, do these imputed phenotypes actually translate into higher power in GWAS? To do this, we need to run a GWAS on the imputed lifetime MDD phenotype and ask if they give more GWAS hits. So first, for reference, I show you again the GWAS on observed lifetime MDD. There's one GWAS hit, and the sample size is 67,000, so not a huge amount of power. Now I show the results from SolveImpute. On the left, I show the GWAS results from just the imputed people. So these are the 267,000 people who are originally missing for lifetime MDD, and we have imputed those values from the MDD-related phenome. We got 13 GWAS hits in this case. On the right, I show the GWAS on the observed plus the imputed lifetime MDD measures combined, and that gave us a total of 26 GWAS hits. So it seems that imputation is improving GWAS power, and by more than tenfold. Now I'm showing you the results from autocomplete in the bottom row. And for both imputed only and imputed plus observed phenotypes, autocomplete does better than SolveImpute in terms of number of GWAS hits you get. So autocomplete improves GWAS power even further. But now the question becomes, how do we know if the new GWAS hits were any good at all? Like maybe we found a bunch of garbage and none of it is relevant to depression. So we therefore did a bunch of downstream analysis that assessed the genetic architecture of the imputed phenotypes, as well as how well they predicted MDD in both UK Biobank and some external cohorts. So first, we asked if the association effects in the GWAS on imputed lifetime MDD were different from those in the observed lifetime MDD GWAS. So using a random effect meta-analysis to test for heterogeneity between the two sets of effects genome-wide, we found there's no significant evidence of heterogeneity between the two sets of analysis at all. In other words, imputed lifetime MDD measures gave similar GWAS effects as observed lifetime MDD measures. There's no evidence of heterogeneity. Second thing we did, we asked if the effects from the GWAS hits on imputed lifetime MDD phenotypes replicate in external cohorts of depression. So this would include, should try to use my pointer. So this includes the 29 core cohorts of the Psychiatric Genomics Consortium. All of them are clinical cohorts collected through structured interviews. This also includes iPsych, which is the can't see my mouse. iPsych, which is the Danish health registry, and that is a doctor's diagnosis of depression, as well as finally the self-reported depression in 23andMe. So in all cases, the effect sizes found in GWAS on imputed lifetime MDD were in the same direction in all of these cohorts, and in many cases they were also significant. So at least we know that they are relevant for depression. And finally, we assessed the genetic correlation between the GWAS on imputed only and imputed plus observed lifetime MDD with external cohorts. On the very left of this slide, I show you the genetic correlation of observed lifetime MDD with PGC, iPsych, etc. And so what we want to be seeing from the imputed ones is that the pattern matches lifetime MDD. And as you can see from this slide, they do match. They look very, very similar. So through this, we can tell that the genetic architecture of imputed lifetime MDD is very similar to that of observed lifetime MDD. The ultimate test of this, of course, would be whether imputed lifetime MDD is able to predict depression either in sample through cross-validation or out of sample. I'll get to that in a minute, but because before that, I need to tell you something else. We need to acknowledge that imputation isn't the only way to integrate multiple phenotypes to improve power. There are other approaches as well based on combining summary stats of genetically correlated traits. For example, there's this method called MTAG, which is a meta-analysis that weighs input GWAS summary stats by the genetic correlations between the phenotypes that are input in this method. And it outputs a meta-analysis summary statistic for each of the input GWASs. In their paper, they use MTAG for depression, neuroticism, and subjective well-being. So for depression, MTAG performs a weighted meta-analysis on its own summary statistic and the summary stats for neuroticism and subjective well-being weighted on their genetic correlation for MDD. So through doing this, they doubled the number of GWAS hits for depression in the process. So obviously, if you're a person and you wanted to be improving GWAS power and you want to be using MTAG, the first question you need to ask yourself is what do you want to put in as inputs? Because it very much depends on what you put in as inputs. And for us, because we wanted to compare MTAG with imputation methods, and because we put in 200 phenotypes into our imputation methods, that would mean we need to put in 200 GWASs into MTAG. And that firstly is quite troublesome to do. It's not impossible. But also, this becomes a problem that starts to not scale like very soon, as soon as we want to be incorporating thousands of phenotypes into imputation. So we think that this is a problem with MTAG, and also there are also other issues with MTAG that may get amplified as you include more input. So instead of aiming to compare MTAG and imputation like to like in terms of number of inputs, we looked into more realistic usages of MTAG, picking 6 sets of phenotypes as MTAG inputs. And you don't need to remember any of this. I'll take you through them as I go along. But what I want you to take away from this table is that with different inputs, we get different mean chi-square values from the MTAG outputs, we get different boosts in effective sample sizes, and we get different numbers of GWAS hits. And that is really easy to come to a conclusion from just this table alone that whatever you get from MTAG is very, very input dependent. And here I show 2 extremes of what we've done. On the left, I show the MTAG run where the inputs are GWASs on lifetime MDD and 3 family history measures of depression. It did not improve power very much. It only gave us 5 significant hits, and most of them are smushed up in the MHC region. So it doesn't look very impressive. On the right, I show MTAG run where the inputs are GWAS on lifetime MDD, shallow phenotyping versions of depression, environmental factors such as Townsend deprivation index, recent stressful life events, etc. And this gave us 30 GWAS hits. So that's very similar to the power gain that we get from imputation. So as you can see, with different types of inputs in MTAG, you get very, very different results. And I'll show you more evidence of this in a minute. But we'll come back to evaluating all of these approaches with PRS predictions now. So coming back to the ultimate test of whether imputation or MTAC is capturing genetic effects relevant for depression, we can ask whether they improve prediction accuracies for MDD, either in sample or out of sample. We first assessed this in UK Biobank using a cross-validation approach. Here I show you the tenfold cross-validation prediction R-square for lifetime MDD in UK Biobank. So lifetime MDD itself is here. It shows a very low R-square. And as you can see, all of the imputation methods in red and the MTAC methods in dark blue gave you higher prediction R-squares. We then asked we could also do this in other cohorts. So we start by predicting lifetime MDD in the non-white British Europeans in UK Biobank, which we didn't use in our primary analysis. And we can see that the combined measure of both imputed and phenotype lifetime MDD always do better than the observed. And also MTAC always does better than the observed. The trend because the ordering of all of these phenotypes on the x-axis is the same throughout all of my plots. You don't need to bother with the individual items. You can just look at the trend, and it's very similar to what I've shown you on the previous slide. We've also shown this in the UCLA Atlas dataset, which is based on electronic health records. And finally, we show that this trend also holds when predicting depression diagnosis in the 2012 and 2015 eyesight cohorts, which are two independent cohorts, both based in Denmark. So that's great. I've shown you now that imputation and MTAC are both able to improve GWAS power and PRS prediction accuracy. They are. Sorry, I have a question at the back. No. I can show you a slide later that talks about that. This is a separate thing. I can talk about it later. Yeah. No. Yeah. I'll show you another slide later. It's not in this talk, but is there any other questions up to this point? Yeah. Okay. Perfect.
So how do you explain that the lifetime MDD does worse than the 23andMe? With the lifetime MDD is the more restricted definition in the UKB, and the eyesight is also. Can you hear me?
Not really.
Okay. So how do you explain that the lifetime MDD is predicting worse in eyesight when it's supposed to be like a more restricted definition? Is it because of the sample size only than the 23andMe? There, for example.
So the question is, why does lifetime MDD predict worse than 23andMe in eyesight when lifetime MDD is supposed to be a better phenotype? And the answer is, like you said, it has a much smaller sample size. We don't have enough power in GWAS, and as a result, you don't predict that well from the PRS derived from those GWAS. Yeah? That's actually the perfect time to be asking questions, because I'm going to talk about something else like right after this. Sorry?
I have a question.
Yeah.
So what's the hypothesis? Is the hypothesis that the people in these larger data sets have multiple different phenotypes? And it's just a GWAS, which is the mixture of all of them? Is that kind of what the hypothesis is? Or is it that some people have multiple things going on?
Right. So the question is, I guess, Zara, you're asking, some of these big data sets like 23andMe, is the problem with it the depression phenotype contains so many different things that is not depression? Is that your question?
Yeah.
Yeah. The answer is probably yes. We think that there's a lot of misdiagnosis in that.
So and many people in there that are not depressed.
Yes.
Right? Yeah.
Yes. That is our general interpretation of it. Okay. So can I carry on now? Okay, great. So so far I've shown you that imputation and MTAC are both able to improve GWAS power and PRS prediction accuracy. But like Zara said, and many people pointed out, shallow phenotyping does exactly that. How is what we're doing different from shallow phenotyping? Yes, we still improve GWAS PRS prediction accuracy beyond shallow phenotyping, But shallow phenotyping is so cheap, it's gonna like reach the right sample sizes very soon, and then why are we even bothering with imputation and MTAG? Because they could just collect more shallow phenotypes. And the reason, of course, is what I told you at the beginning, is that shallow phenotyping has low specificity. But so far, I haven't told you anything about like whether imputation or MTAG solves this problem or not. And of course, it's quite a difficult problem to be solving. The question for us now is whether imputation or MTAG can do better in terms of specificity to depression than shallow phenotypes of lifetime MDD. And in order to answer this question, we first have to quantify the problem. How problematic are each of these measures of depression? So we started doing this by using PRS generated from both imputed, MTAG, shallow phenotypes of depression, etc., to predict other phenotypes that are related to but not depression, such as neuroticism, stress, socioeconomic status as indexed by Townsend Deprivation Index, and one's lifetime experience of trauma. So each of these have some level of genetic sharing of depression and are usually amplified in GWAS, or shallow phenotyping definitions of depression. And as you can see from these plots, the phenotypes are still ordered in the same way as I've shown you before. The imputation and MTAG runs, not only do they improve GWAS PRS predictions of lifetime MDD in various cohorts, they also do that for neuroticism, stress, Townsend Deprivation Index, etc. So in other words, their improvements in PRS prediction accuracy is not specific to depression. And the question really is how not specific. We need to come up with a metric for specificity which can be used to compare the deep phenotyping versions of depression versus the shallow versus the imputed and the MTAG ones. And we thought about this and realized that in order to effectively think about specificity, we also need to think about this other related concept, which is pleiotropy. And hopefully, after a few slides, you see why I'm talking about this now. So pleiotropy, as you guys know, it occurs when one gene influences two or more traits. For a disease as polygenic as depression, we would be expecting that a lot of genetic factors that are influencing it is also going to be pleiotropic to a lot of other traits. So in other words, lifetime MDD itself has natural pleiotropy to a lot of other things. And as such, on the polygenic level, a PRS derived from a GWAS on MDD can be predictive of more than one trait, as we've seen in the previous slide. Hopefully, if the phenotype you're actually using to perform GWAS on is any good, PRS is going to be able to best predict itself than other traits. But if the phenotype that you're using for GWAS is noisy and contains misdiagnosis and therefore spillage of genetic effects from all sorts of other things, it is going to be disproportionately predictive of other traits than of itself. So using this, we can derive a measure of this spillage or non-specificity. And the term that we've come up with is PRS pleiotropy, which is simply the ratio of a PRS's prediction of secondary traits over the actual trait that you're interested in. And in this case, it's lifetime MDD for us. So in this cartoon plot, I show some data points that will make up the PRS pleiotropy distribution of observed lifetime MDD in UK Biobank. So of course, like on the top there, you see that if you took the prediction R-square of lifetime MDD over itself is 100%. That's no brainer. But then, so for example, for these other dots, these two are shallower phenotyping versions of depression. Lifetime MDD would predict them at a lower R-square than itself. And this is neuroticism, and lifetime MDD would also predict it at a lower R-square than itself. And how low this is really indexed by genetic correlation between lifetime MDD and these other traits. So I can perform this analysis on the entire MDD-related genome, not just 4 traits. And if I then also order them by their PRS pleiotropy, I then get a curve that gives me the PRS pleiotropy distribution for observed lifetime MDD. So on this slide, I show you real data. This black line is the actual curve of lifetime MDD. And in the MDD-related phenome that we use for imputation. And for comparison, I have the pleiotropy curve for GP-Psi, the shallow phenotyping version of depression, in gray, and the pink line is that for 23andMe, which is the self-reported version of depression. So as you can see, the black line for the observed lifetime MDD, the deep version of depression in UK Biobank, is lower than that of GP-Psi. And that's what we expect, because GP-Psi, being a shallower phenotype than lifetime MDD, would have higher pleiotropy to most of the 200 phenotypes in the MDD-related phenome, and is therefore less specific to lifetime MDD. And 23andMe is very similar to GP-Psi, having been collected in a very similar way. To make this even clearer, we can take the PRS pleiotropy of observed lifetime MDD as a baseline and subtract it from GP-Psi and 23andMe to arrive on the plot on the right, and where we're just plotting excess pleiotropy. So the excess pleiotropy plot really tells us how non-specific a PRS is. The higher these lines are, the higher these lines are from the black baseline, the more non-specific the PRS is. So having seen this done on GP-Psi and 23andMe, we can now apply this to both PRS from the imputed and the MTAG outputs. So on the left, I show you the results from MTAG. And there are several interesting things on this plot. So firstly, the dark blue line, the topmost line on this plot, is the MTAG run with environmental factors as input. So these are environmental factors including education attainment, Townsend deprivation index, etc. And as you can see, it has really high excess pleiotropy. It shows massive spillage from phenotypes such as education attainment, whether you smoke, the maternal smoking at birth, etc. So this shows that it is very non-specific to lifetime MDD. So even if this MTAG runs gives us a lot of GWAS hits, we may not be wanting to use it if our interest is to find genetic signals specific to depression. The second thing that is interesting is this steel blue line, the lowest line on this plot. And that represents the MTAG run with family history data on depression as input. This shows really little excess pleiotropy. In fact, it's even less in excess pleiotropy than lifetime MDD itself for unrelated things such as college, whether you've gone to college, whether you smoke or not. So even though this doesn't give us that much of an increase in GWAS power, maybe this is what you want to be using if you want to be identifying specific GWAS hits for depression. And finally, this line here is the MTAG run with both of those inputs combined. And it lies somewhere in the middle in terms of specificity. So as comparison, I show results for soft-impute in purple and autocomplete in brown, I guess, it appears on the screen. You can see that the purple line for soft-impute is very similar to that of this line, which is all of the inputs in MTAG. And they both consistently lower than the gray line for GP-Psi, which means that it captures less pleiotropy than the shallow phenotyping version of depression available in UK Biobank. So not only are these able to give you more GWAS hits than the shallow phenotyping, they're also more specific. So if you're looking for maximizing both GWAS hits and specificity, you want to be using these approaches rather than just the shallow phenotype itself. And the interesting thing is what's difference between this line and this line here is that the purple line is a result of drawing from more than 200 phenotypes, while this one drew from around 16. So if you're thinking about improving GWAS and GWAS power and specificity with a relatively agnostic approach that doesn't need you to be very knowledgeable in terms of what inputs to put into MTAG, then maybe soft-impute is your best bet. So I've come to the end of my presentation. In summary, what we've done is we've looked at power specificity trade-off between deep and shallow phenotyping in depression, which is a very well-established problem. We are trying to find a way to make use of both types of phenotype, and that seems like a good idea to utilize this in biobanks and electronic health records. And we've shown that imputation and MTAG both improve GWAS power. Both of them also can improve PRS accuracy. Sometimes MTAG can do better than imputation. However, the specificity of PRS is preserved better by soft-impute and some choices of MTAG than others. There's a lot of future work in this area. We need to refine the pleiotropy measure a little bit more. It's not really currently a real method. It works for what we are doing now, but it can definitely be done better. Also, we want to be incorporating more types of data. At the moment, we are using 200 phenotypes from UK Biobank. We can definitely include other things like imaging, omics, etc. And that would involve scaling up the methods as well. And finally, we want to be improving portability across cohorts and ancestries, which is something that I haven't talked about today. Thankfully, we got a grant to do all of this. So if you want to come and work with us, this is the people who I work on this with. We have positions in my group, in Andy's group, in Andrew's group, many other groups. So if you are interested in doing this kind of work with us and potentially also working on depression heterogeneity as specificity later, please come and talk to any one of us. Thank you very much, and I can take questions now.